well. And part of this relates to variants of unknown significance. And so the title of my talk is Variants of Unknown Significance. What do they mean? And I hope to let everyone fully understand what it means whenever a genetic test result has that and how that relates to us understanding diagnosis. I want to bring up one point, and I've been trying to do this in all of my talks, is to really elucidate even how the inclusion of indigenous people and underrepresented minorities is important in all aspects of research, just not clinical trial development, but even in genetics. And in order for us to really understand variants of unknown significance, it's important that we have genetic data not just on individuals with disease, but also individuals without disease, and that these individuals without disease are from a broad background. And so one of the programs the NIH has is called All of Us, and I encourage you to look into it, but it's a way of broadening much of the genetic databases so that it's inclusive not just of Europeans and U.S. citizens that are predominantly white, but also those of underrepresented minorities and indigenous people. So I'll start with just kind of the framework of genetics, which is really that we have a mutation or a gene, I guess I should say, in DNA. So this represents a gene in DNA. So DNA is a double helix. It's the one copy of one of these strands is from your mom, and the other copy comes from your father. And this gene is transcribed into an RNA, and that RNA is then the recipe that then gets created into a protein through a process called translation. And just so that we're all kind of understanding on the same level, we can think of DNA as being kind of the cookbook, and each one of these tabs represents a gene or a recipe. And just as you might have this cookbook that your grandmother may have, and she then sends out recipes, she doesn't just give one recipe out, she gives many recipes out to many different people in the family, and that's how RNA is. It's transcribed multiple times, and that allows us then to make the recipe, in this case, a cake. And sometimes what happens is we get a variant or a mutation in this recipe that makes us make a different type of cake. And I want to point this out because we didn't not make a cake. We still made a cake, but in this case, the cake looks a little bit different than the previous cake. What I don't know from looking at this cake is, does it taste the same? Is it really, can we really still call it a cake? And so that's some of the areas that we think of with variants that happen in disease genes. And so I bring up again this recipe here, and the way we think about genetics is I can have mutations that are created in this recipe. And so you can imagine this recipe here. You can imagine that we deleted all of the sugar and all of the cocoa, and then we did the recipe. And you and I pretty much know that that's not going to be a cake. That's going to be a pretty terrible cake. And so that's where I'm pretty confident that I could interpret that that cake is not going to be the right cake. However, what if this part's deleted? What if I forget to preheat the oven? I just turned it on to 145 degrees. I'm probably still going to get a cake, and it may taste almost the same. And so that's a mutation that's happening in this recipe that is a variant that might be tolerated. Another example is, well, maybe there's a mutation in this recipe that gets rid of buttermilk and just leaves milk. I'll probably get a cake. It may taste different, but I guess that's my point, is that sometimes I don't know. What if I baked this cake for 120 minutes? I don't actually know. I might still get a cake. I might still get a pretty good tasting cake. And so, or I might get something that's burnt. And so the point is that we're trying to make predictions in the recipe and trying to understand whether that prediction or that variant is going to cause a problem in the protein being, in this case, a cake. And so whenever someone gets genetic test results, often we get mutations in the recipe. And so in this case, in this gene here, AMPD1, there are a mutation or a protein change that's occurring in that gene. There's other genes as well. And how do I know that this mutation here didn't just change the flavor, but I'm still getting the protein? Or how did I know that this one here isn't completely causing a non-functional protein? And I can't tell that just from looking at genetic data. And so that's the framework with which I'd like to think about genetics and variants of unknown significance. And so the goals of what I want to talk about are types of genetic testing, barriers to genetic testing, understanding what I just said, which is genetic variation, variations that happen in the recipe, and then how do we interpret those variations and interpret genetic test results. And so genetic testing performed in clinical practice is done in a couple of different ways. One is individual gene testing, and that can be appropriate if a family member is already diagnosed with a form of limb-girdle muscular dystrophy and they know that they have a mutation, and that would be an appropriate way to do that test. Typically, what we do are panel-based gene testing, and that's genetic testing for all the known or likely limb-girdle muscular dystrophies. There are different panels offered from different genetic testing companies. So if someone says that you had panel-based testing, your panel could be different than somebody else's panel. In addition, depending on when your genetic testing was done, older testing panels have fewer genes than newer panels. And so it may not be inappropriate if your doctor says, I don't know what type of muscular dystrophy you have, to repeat a gene panel, because the gene panels have expanded and they've identified more and more variants in different genes. Another approach is whole exome sequencing. And this is an approach that sequences every known gene in somebody's DNA. And that can be appropriate as well, but part of the problem with doing a whole exome sequence is that you're going to identify mutations in genes that are unrelated to limb-girdle muscular dystrophy, or at least known causes of limb-girdle muscular dystrophy. And that can actually add more confusion sometimes, in which case panel testing may be appropriate for first line. And finally, there's something called whole genome sequencing, and that sequences the entire genome. That's something that can be done as well. It can be challenging to interpret. Part of the reason it's challenging to interpret is because it is so much data, and it can be often even hard to handle that data. So when I think of genetic testing, I think often about some of the barriers to genetic testing and diagnosis. And some of these barriers are in our control and some of them are out of our control. And so one of the barriers is payers. So will a insurance company pay for genetic testing? This has been actually almost abolished because of free genetic testing programs. And free genetic testing programs have really taken that barrier away. Another barrier is patient knowledge. Patients don't realize that they might need genetic testing. They might have been told they've got a form of limb-girdle muscular dystrophy, but they've never had genetic diagnosis performed. And so that's another where patients and even patients' family members need to be educated on the importance of genetic testing. You might say, how is that a barrier? Well, if I can't get a patient to get genetic testing done, then I can't do genetic testing. So that can be a barrier. Community knowledge, and this conference is great, and this opportunity to talk to you is great because I can educate the community that making sure you have accurate and recent genetic test results. I advocate that patients have their genetic test filed away just like they have their Social Security card filed away so that they know exactly what their genetic mutation is. Physician knowledge. Not all physicians know about genetic testing and what needs to be done and whether a different panel is different than another panel. And so I think educating physicians is something that's really important. And finally, I think this one is the most interesting, is patient care guidelines. And actually the most recent evidence-based patient care guidelines in 2014 actually have different levels of evidence. And so level B evidence is via a cohort of different physicians say that it's a good idea. Level C evidence is that it's an expert opinion. So basically my idea that it should be done. And the importance about this is that level C evidence suggests that exome sequence or even panel-based sequencing be done. And so we really need to change those guidelines because that's not expert opinion anymore. It's really should be standard of care that if you don't know the genetic testing of a patient, that you should do either exome sequencing or panel-based sequencing to identify the genetic abnormality. So in thinking about breaking down test barriers, I really give credit to the Jane Foundation as well as the Muscular Dystrophy Association, where they started offering free genetic testing programs in MDA clinics. And I think this is a great figure where they're able to show the breakdown of the different types of muscular dystrophies that occurred in patients seen in these clinics. And we're able to show the percentage of patients that have this. But this figure here, which is the one that many of us see, is only part of the story. The actual story is that only about 23% of patients fell into that category, and that 77% of patients actually are undiagnosed, and half of them have what we call variants of unknown significance. And so how I think about it is that we broke down some barriers, but we revealed some new barriers and some new challenges to genetic test results. And so what are the challenges of variants of unknown significance? And those are the variants or the changes in the recipe that lead us to not be able to predict whether that cake or that protein is functional or non-functional. And the challenge is that it leaves patients with diagnostic uncertainty. So patients don't know, do I have this disease or not? It allows for the inability to really start to plan for your family and say, I think that this could be inherited. I want to have kids. I don't know. So that diagnostic uncertainty is really important to resolve. It leaves physicians with diagnostic uncertainty. And I put that up. Not that we're the most important person, but we really do care to make a diagnosis, and it's frustrating for physicians not to be able to make a diagnosis on the patient's behalf. And most importantly, it may lead to inappropriate treatment and other testing. If I can't tell you that you have a certain disease, you may undergo multiple tests, biopsies. God forbid there is a therapy that you get and it's an inappropriate therapy. Even thinking of a gene therapy approach that could be, even if you have the disease and you don't get the gene therapy approach, but let's say that I tell you you do have the variant and it's causing the disease and you get the gene therapy inappropriately. So that's equally important. So making sure patients do and don't have the different limb-girdle muscular dystrophy disease genes is important. And ultimately, it also leads to increasing healthcare costs and patient frustration. I think this is important too, because we haven't resolved many of these variants. We start to not understand what the true prevalence is of many of these limb-girdle phenotype subtypes are. And I think that's really important because the way that we start to understand resources and engaging drug companies is by making sure they understand how common or uncommon certain subtypes are. And I think I already touched on this. You might ultimately miss out on a certain therapy if we can't make your variant known as being pathogenic. Again, the same thing being said again in a different way. We want to variant resolve because we want to resolve pathogenic variants to determine if people are amenable to therapies, but we want to resolve ones that aren't pathogenic, benign variants, because we want to make sure that a future therapy is potentially contraindicated. And one thing I can also say is by resolving a variant in you as a patient is actually improving patient care for other patients that may have variants or similar variants. And so as we start to do this, it's really something where you as engaged patients can help make a difference for patients in the future or patients that might be less engaged in their care for a variety of different reasons. So it wasn't really until 2012 that we really appreciated the amount of human genetic variation. And it was this this paper here, which was from the 1,000 Genomes Project. And I always think it's funny because it's 1,000 Genomes Project, but they did 1,092 human genomes. But it was called the 1,000 Genomes Project. It was really the first description of what we call rare variants. We already knew that there were variations in families. We'd already been doing that type of studies, in crime labs to understand, even paternity testing. We knew what we call kind of common variation, and if we looked at multiple common variants, but what we didn't know was the amount of rare variants. And what became interesting is that for a single individual, there are many common variants, but there's a small fraction of rare variants, and there's even new mutations or new variants in everybody. And so the way I think about this is that everyone has 3 to 4 million variations. These may be common, but 30 to 40,000 are rare, and some of them are unique to you. And that's not just patients with limb-girdle muscular dystrophy. That's everybody. That's me. That's you. That's everybody in the room has 30 to 40,000 rare variants. Of these rare variants, 250 to 300 are loss of function variants, meaning that I can predict that they're going to cause a non-functional protein. That's amazing to me, that within our human variation, that we have mutations in genes that cause them to be detrimental. 50 to 100 of those variants are in genes implicated in inherited disorders, and 0 to 7 of them in everybody. So some people have known pathogenic variants in genes but don't manifest with disease. And so I think it's really, really interesting. And so to me, it really should be no surprise that whenever we do a genetic test, that many of us have variants of unknown significance, because as I said, we have 30 to 40,000 rare variants. Some of those are going to be in limb-girdle muscular dystrophy genes. The other way to think about it is whenever we do genetic sequencing of patients, and I actually put here actually of any individual, as we go and we take those variants. So we do a patient or even myself, I have 2 million variants, because I just told you that everyone has 3 to 4 million variants. So let's say I have 2 million variants. We then do a filtering process where we look for the ones that are really rare. And so now we end up with that 30,000 to 40,000. We look for ones that actually might be deleterious. We look for ones that we think could functionally predict. And now we're up to 3,000 genes. That's still a lot of variation in genes that could be causing disease. And then we try to enrich for variants that might track with disease. And the point is that everyone has variants of unknown significance, up to 3,000. And so whenever we look at genetic test results like this, and we see, and they've got variants, I very well could have this exact same pattern on my test results. And so the question is, does the patient have progressive weakness, and are these pathogenic or are these incidental? And as we look through these different variants, here's another patient's genetic test results where they have variants within genes and multiple genes, and are these pathogenic or are these incidental? And so this leads to kind of a detour in how we could do genetic testing. And I think of two different approaches. There's a gene first approach, and where we do the genetic testing first, and then there's clinical pathologic assessment. So see the patient, do a history, do laboratory studies, and do that first. And we're going to give both examples, but there's advantages to both. The ease and the low cost of genetic testing has led many patients to receive genetic testing first without clinical assessment by a neuromuscular physician. And I think we need to acknowledge that that's a reality that's happening, that in many cases, gene testing is being done first, and we could imagine a day where gene testing will be done before anyone even sees a physician. And so that's one approach. The lack of experience with genetic testing leads many neuromuscular physicians unable to appropriately order and interpret genetic testing. And so that's the example of going and seeing a clinician, and the clinician sees you, does a clinical pathologic examination, but doesn't really know what tests to order and how to order them. And so I think my point is that we need to be working together. The geneticists who want to order the genetic test first need to be working with the clinicians who want to examine the patient first. And so that's the goal in my mind. The vision is that we not put it on an individual caregiver or physician or geneticist, but that we work together. And so I want to give an example. This is a patient of mine who came into clinic, and he'd already had genetic testing done. And in the case of his genetic testing that was done, he was identified as having a variant in a limb-girdle muscular dystrophy gene that was identified. It was pathogenic. It was in other family members. He had a disease that made sense. He also had a variant in another gene, but that gene was a variant of unknown significance, and he had already a slam dunk made. So he already had a genetic diagnosis whenever he showed up to me, and that was done. This is another patient that showed up to me. This patient showed up to me and said, hey doc, I have limb-girdle muscular dystrophy because I have a mutation in this gene. He has a variant of unknown significance. As you can see, he was a male with developmental delay. He didn't even have muscle weakness. And so my point is that just because you have a genetic test that shows a mutation in a gene, it doesn't mean that you actually have the gene. The geneticist, and so this patient was never seen by a physician. He was seen only by a geneticist. The geneticist can tell you if that gene is rare and unique. They can offer some possibilities about whether it's deleterious. But unless that variant has been identified as pathogenic and is put into a clinical database, it will be a variant of unknown significance. And so this patient needs to be seen in clinic to try to understand if he has a variant, if this variant is pathogenic. Again, I want to give more examples where it just becomes really diagnostically challenging. I don't want to harbor on all of these, but you can see that everybody has variants of unknown significance in multiple genes. This is not just a patient with limb-girdle muscular dystrophy. I want to focus on this example here, where this patient is a 34-year-old man with limb-girdle muscular dystrophy of unknown genetic cause. He had a panel performed, and he was identified as having a pathogenic variant in one gene. And so for some diseases, you need to have two mutations or two variants. For some other diseases, you just need one variant. And we'll talk about that a little bit in a minute. But in this case, for calpain 3, limb-girdle muscular dystrophy 2A or R1, you need to have a variant in two different genes. And so he had a pathogenic variant in one gene, and he had a variant in another gene. And this one was an interesting variant because this variant didn't change the protein. This was one where I couldn't look at the amino acid change and tell you if it was going to be a good cake or a bad cake. In fact, if I were to look at this, I'd say this isn't going to change the cake at all, is how I would look at this without doing some more work. And so this patient had a variant of unknown significance in this gene. So how do we decide? How do we resolve the variants? What are the steps toward a diagnosis? Well, we try to do what I call an integration of the genetic data and the clinical data. And so we take a patient history. We find out when the age of onset was. We ask what the first symptom was. We see if they have other symptoms, maybe skin changes, heart problems that might make me think of other diseases. Do they have a history of needing a cane or walker in the family? I ask every patient, some of these diseases are late onset, and I find out that the father died at the age of 28 because of a car accident. Well, that's helpful to know. He may not have had muscular dystrophy, but he may not have been old enough to manifest with weakness. Cousins with muscle weakness, that can be helpful. So thinking about distant family members. Physical exam, does he have weakness, contractures, other features? In the case of this patient, his weakness began at age of 25, and his first symptoms were difficulty climbing steps. It's really helpful. That tells me that his weakness is proximal, what I would expect for someone with limb-girdle muscular dystrophy. He didn't have any family history of weakness, not uncommon in genetic diseases. In the United States, we have small family structures. We don't always know our extended family very well. And so no family history of weakness does not mean that it's not a genetic disease. Proximal weakness was scapular winging, so he had a pattern of weakness, and he had this kind of interesting aspect where he had weakness in his shoulder girdle that caused scapular winging. That can be helpful as a clinician. Other aspects we look at, interpretation of laboratory data. So muscle enzymes, electrodiagnostic studies. I would say every patient who could have a muscular dystrophy, we want to make sure they've had an EMG done. Electrocardiogram, do they have heart issues? Sometimes an MRI is done, and often a muscle biopsy is done. In the case of this patient, his muscle enzyme was elevated. It was 2200. He had electrodiagnostic studies that showed that he had a myopathy. He didn't have something like a neuropathy. You can see how this myopathy, this EMG is not telling me anything other than this patient has a muscle disease. It's not telling me what the genetic cause is. We did not do an MRI on this patient. He did have a muscle biopsy. His muscle biopsy showed chronic myopathy with muscular dystrophy. Again, helpful for me to know that he had a muscular dystrophy, but nothing specific telling me that he had a certain subtype. We then go and we become geneticists in some ways. We do database searches. We do functional predictions on those mutations. And remember, he had a mutation in calpain-3. He had 2 of them, and he had one in dysferlin. The calpain-3 is unreported in population databases. With a little bit more legwork, you can actually see that it's very close to what we call a splice site, leading to what's called a donor loss. So it could lead to a truncated protein. That dysferlin variant, although he only has one dysferlin variant, is rare, and it has a minor allele frequency of 0.03. This is one of those 3,000 rare variants that this patient probably has that may or may not be pathogenic in this patient. The other things we do is we say, well, let's expand the patient's pedigree. Are these mutations on different chromosomes? I don't know that from just doing a single patient. And so we expanded the pedigree, and we found out his father was actually had already passed away, but his mom had one of the mutations in the calpain-3 gene. His sister was a carrier, and the other sister that was unaffected. So these were unaffected patients, and it would make sense they were unaffected because they didn't hold both those variants and both those mutations. We were able to do immunohistochemistry on his muscle biopsy. The immunohistochemistry, in some cases, this showed abnormal dystrophy-related protein stains with reduced dysferlin. Well, that makes me start to think that maybe this patient doesn't have mutations in calpain, but actually has a mutations in dysferlin. We need to go the step further because he only has one mutation in dysferlin. And what we do is, in our lab, we do an extra step where we actually do a Western blot to actually look at how much calpain-3 protein is there. And you can see this patient has absent calpain-3 protein, but normal levels of dysferlin. And so in this case, we solved this variant is resolved. This patient has limb-girdle 2A, R1 calpain-3 associated. And as I hope you can see, this was time-consuming. This took specialists. This took me thinking about this patient. This required acquiring patient family structures and DNA. This required specialty testing. And that's what it takes sometimes to resolve these types of variants. And I can't expect non-specialists, and I can't even expect some neuromuscular physicians to be able to do this process. And so I think it's really important to appreciate that. I bring this up because Brad Williams asked me to make a comment on this, and he said, you know, some limb-girdle genes are even more complicated. And so I put here the complicated becomes even more complicated. And so some limb-girdle muscular dystrophy genes can cause a disease in a recessive manner, meaning that you need 2 disease mutations, one from mom and one from dad. Some limb-girdle muscular dystrophy genes cause a disease in a dominant fashion, meaning that you just need 1 disease mutation, and you can get that variant from mom or dad. But the complicated gets more complicated because we're learning now that some limb-girdle genes can be inherited in both manners, in a recessive or a dominant fashion. And it makes the challenge of variant resolution very complicated. And I don't have a clear answer for how to solve that problem. And likely it's going to require just more sequencing of patients and families, and more looking into variant and mutation-specific variation in patients. And so hopefully the process I'm going to describe of how we'd like to look at more variation will help. We've become to be part of a group called a ClinGen. This is an NIH Supported Infrastructure. It does exactly what we said. It's systematic variant resolution. And so it integrates patients, clinicians, genetic testing labs, and researchers to really ask three fundamental questions, which is a gene associated with the disease? Is that variant causative for that disease? And really the one that I think will become the most important is that information clinically actionable. And we think of this in cancer genetics, where if someone has a BRCA1 variant that's known to be pathogenic, that they need to be actionable. And hopefully someday, if you find that you have a variant in a certain limb-girdle muscular dystrophy gene, this will be a clinically actionable, meaning that you will need to know that there's a therapy out there for that disease. And the way that we do this is by a very complicated approach where we look for levels of evidence and try to integrate in population data, functional data, segregation data, all the stuff I just described to you, and try to do that in a systematic way and kind of build levels of evidence and strength. And I like this slide because they're Lego blocks, and what we're trying to do is build them and stack them and actually have variant classification and ultimately lead to what we call expert panel reviewed. And so I finally just want to summarize here that at present, genetic test interpretation, or what I call armchair variant resolution, really requires knowledge by both clinicians and geneticists in muscle disease. Systematic variant resolution, which is what we'd like to do, is really, I think, the way to go, so that basically we can move into a gene-first approach where anybody can get genetic test results and they'll know if it's a pathogenic variant. And finally, what I call enhanced clinical phenotyping, it needs to be done by a limb-girdle muscular dystrophy expert to really understand a genetic diagnosis, and that integrates all of these aspects into kind of this stacked level of care. And I want to thank members of my research group. I want to thank a large LGMD research consortium, which is helping us understand these questions as well. So thank you.